Hello, my name is Anton Mahama, and I'm from Oakland University in Michigan. And I'm a master's student, and I'm working with Dr. Fabio Battistuzzi on domain signatures of genome complexity in extremophiles. So what are extremophiles? Extremophiles are those interesting and unique organisms that live in extreme environments. And they live in hot and cold niches, and they also live in acid solutions, alkaline solutions, salty environments, and they include members of all domains of life: Archaea, eukaryotes, and bacteria. And Archaea is the major group that survive in extreme environments. And although members of this group are less versatile than bacteria and eukaryotes, they're quite skilled at adapting to extreme conditions. And extremophiles exist in all of the archaeal phyla. In our data, we only have the Cren Archaeota, the Uri Archaeota, and the Thumb Archaeota. And these are some examples of the extremophiles. We have the hyperthermophiles in red, and then the thermophiles in green, and then we have the psychrophiles in blue, and then piezophiles, also called barophiles, in black. And as you may know, that the extreme environments are unstable environments, so that they're likely to lead to high variability at the genomic level. So any sort of adaptation or surviving strategy is likely to have genomic signatures. And here we're looking at these genomic signatures in the organisms, and part of these genomic signatures, which are part of the bigger group, are LCRs, the low complexity regions. And LCRs have been known to be evolutionarily significant because they provide genomes and individual genes with adjustable turning knobs for efficient adaptation. So previous research has been done by Harty and Golding, and it showed that LCRs are not conserved regions and that they accumulate mutations. So here at number 0, we have the LCRs, and these are the number of mutations they accumulate, the proportion of SNPs, the single nucleotide polymorphisms, and the different colors here in the graph shows the different conditions they tested the LCRs in. And genomic variability has been tested in eukaryotes and in viruses, and in the viruses it was suggested that LCRs are an important source of genomic variability for the lentivirus, and that they're an important source for antigenic variability in the HIV populations. So what are the low complexity regions? The LCRs are repetitive sequences that contain few nucleotides or amino acid types, and some regions contain few different nucleotides or amino acids, while others only contain one. And LCRs have various configurations, ranging from periodic to aperiodic motifs. And they have different nomenclatures ranging from low complexity sequences, tandem repeats, simple sequence repeats, amino acid repeats, but they all basically share the same low complex or low diverse sequence. So LCRs are ubiquitous in eukaryotes, but still their function and the evolutionary forces that act on these regions are unknown and are poorly understood. And here on the right, we have examples of LCRs in different eukaryotic species. So for example, in the Homo sapiens, the humans, we have about 18%. And then in the Plasmodium falciparum, the malaria agent, we have about 50%. So the proportion of genes that contain LCRs is highly variable across the different eukaryotic species. So all the information that we have so far on LCRs is a result of research on eukaryotes. And here you can see the eukaryotic tree, and the parts that are marked with a star are the ones that have been studied for LCRs, and even those form the minority in comparison to the other eukaryotic species. And no one actually has looked at prokaryotes for LCRs, and although the prokaryotes form the majority of the fully sequenced genomes, thus they provide us with a huge set of data to analyze and work with. And prokaryotes also form 2 out of the three main domains of life. And all the analysis that has been done so far was done on model eukaryotic species. So in order to study a full domain, we need to develop new testing methods and analysis in order to automate the process. So we decided to focus on Archaea in our research because Archaea has about 200 species, while bacteria have 13,000 species. So when we study Archaea, we will be able to develop these new testing methods and to automate the process in our future research. So what we actually did, we got the fully sequenced genomes of Archaea from NCBI, and it was about 221 species. And then we did basic statistics to see the LCR patterns and trends in the Archaea. And at that point, we don't really know if LCRs even exist in Archaea. So now I'm going to be talking about the LCR patterns that we explored in Archaea, and the LCR frequency, LCR length versus protein length, the LCR frequency versus the GC content, the amino acid composition and usage, and the LCR location in the proteins. So to calculate the LCR frequency, we just followed a simple equation, a number of proteins with LCRs over the total number of proteins times 100. And on this tree, you can see the red are the temperature extremophiles, and then the yellow are the temperature mesophiles, and the orange bars are the LCR frequency. So on average, the LCR frequency ranged between 3 and 7%. And when we looked specifically at certain groups, for example, the Halobacteria, the salt-loving extremophiles, we saw that they have the highest LCR frequency, which was about 14%. And Halobacteria are salt-loving extremophiles. They live in salty extreme environments. And the red water here shows the Halobacteria. And then we looked at other groups, such as the Archaea, which are temperature extremophiles, and to be exact, hyperthermophiles. And they also have a high LCR frequency, but it's lower than the Halobacteria. And theirs range between 10 and 12%. So we compared the LCR frequency between these groups to see if there's any significant difference. And as you can see, we saw that the Halobacteria have a higher LCR frequency than the Archaea, and they have a higher LCR frequency than the Archaea. And the Archaea in this case includes both extremophiles and mesophiles. So the main question here is this high LCR frequency a characteristic of salt extremophiles or temperature extremophiles or extremophiles in general? So to answer this question, we did a further analysis comparing the LCR frequency between Halobacteria and Archaea. And as you can see, we saw that Halobacteria has a higher LCR frequency than Archaea in general. And even when we compared the mesophiles and extremophiles in the Archaea, we see that the mesophiles have a higher LCR frequency than the extremophiles. So this suggests that the high LCR frequency is a characteristic in the salt-loving extremophiles. And there are actually few possible explanations to this trend that we just saw, and one of them is that LCRs are an emergent property of proteins, meaning longer proteins will have longer LCRs, and when we compared them, we didn't see this correlation between them. So if the LCRs are not an emergent property of proteins, the LCRs might be driven by the GC content of the genome, whether it's high or low GC content. So previous research done by Tian showed that the LCR frequency is driven by the AT content of the genome in eukaryotes. And in our data, as you can see, the Halobacteria in the yellow, temperature extremophiles in blue, temperature mesophiles in red, they all follow the same trend. And when we did a comparison for significant differences, we saw that the Halobacteria has a higher GC content than all of the other groups, the CREN, the URI, and then all of the Archaea in general. And even the mesophiles had a higher GC content than the extremophiles. So that means, yes, the patterns of GC and LCR frequencies are the same, and that the LCR frequency is driven by the GC content of the genome. Then we looked at the specific amino acid usage between extremophiles and mesophiles, and we saw that they both use GC-rich amino acids. But extremophiles also use AT-rich amino acids, which is expected, as we saw they have a lower GC content. And when we looked specifically at the Halobacteria, we saw that they use similar amino acids to the mesophiles, and they are GC-rich amino acids. And then we compared the amino acids usage between the Halobacteria and the Archaea, and we found that they have different amino acids in their composition. And actually, previous research in eukaryotes showed that the amino acid composition is species-specific, and it's different between the groups. And as you can see, these are 3 different research done on vertebrates, Drosophila, and Plasmodium falciparum. And if you look closely, all of them use different amino acid composition in their LCRs. And the reason actually behind this difference is still unknown. So up to this point, just to summarize, we saw that the LCRs are driven by the GC content of the genome, and the amino acid composition confirmed this, as we saw that they use GC-rich amino acids. So this suggests the neutrality of these regions and that they are functionless regions. So we decided to look at the LCR locations in the proteins because this might be a sign of potential selection. So we followed a previously developed methods where we divided the protein into 3 segments, the N-terminal, the middle, and then the C-terminal. And then we calculated the difference between the observed distribution of the middle point of the LCRs and the expected distribution to locate where each LCR sequence would fall. And our null hypothesis suggests that the LCRs are randomly distributed, and our alternative hypothesis suggests that they are not randomly distributed, and that means that there's a potential biological function for them. So the results of our chi-square shows that in extremophiles we have 82%, and in the mesophiles we have 75% significance, which means that the LCRs do prefer certain locations and that they're not randomly distributed. And when we look at the extremophiles and the mesophiles, we see that they both follow the same trend, where the LCRs are located at the termini of the proteins. And when we look at the Halobacteria specifically, we also see that they follow the same trend, and the LCRs are at the termini of the proteins. And this actually follows a previous trend that was found in research in eukaryotes, and this one specifically in the Drosophila, where it shows that the LCRs prefer to be at the extremities of the protein rather than in the middle. This suggests that there might be a potential function for these regions and that they're not functionless regions. So do LCRs actually help extremophiles to survive? Well, maybe, but we do know that LCRs are a common feature in Archaea, and that they exist in salt-loving extremophiles in a higher frequency than the other extremophiles. We also saw that the LCRs are driven by the GC content of the genome, and this was confirmed with the amino acid usage, as we saw that they use GC-rich amino acids. And we also saw that the amino acid usage is species-specific and different between the groups. So all of that, with the non-random distribution of the LCRs, we can say that the LCRs may be creating genomic variability for the Halobacteria, the salt-loving extremophiles specifically, so they can survive in their extreme environment. But we did not really see this trend in the temperature extremophiles, so this suggests that they may be using different adaptive strategies to survive in their extreme environments. So I'd like to thank Oakland University Center for Biomedical Research, NASA, NIH for funding this program. And I would like to thank Dr. Shailesh Lal, Randall Westrick, and all of my lab members for helping me in this research. Thank you for your attention.
All right, thank you, Anton. So we're running a little bit short on time, but we have time. I'm sorry. We have time for one quick question. So, great.
Hi, thanks for the good talk. It's nice to see someone here at this conference that cares about Archaea. My name's Tian from the University of Connecticut, and I was wondering if you looked at the distribution of other Archaea halophiles and what the distribution of the LCRs were in those other halophilic lineages.
We looked specifically at the fully sequenced genomes. So these were the only ones that were available on NCBI to look for their distribution.
There are like 2 or 3 other halophilic lineages with like really weird GC content, so I was just curious.
Well, there was actually a paper back in 2008 that explained the high GC content in the halophiles, and that convergent evolution have to do with this weird GC content in the halophiles specifically, and not the non-halophiles. Thank you.